Horizon Discovery でございます。遺伝子情報に応じた個別化医療実現のためのゲノム編集の活用。セールスおよびコマーシャル担当のバイスプレジデントでございます Mr. カム・ダリワルさんです。
Thank you. It's a pleasure to be here today in Osaka once again. I'm going to talk more about drug discovery, primarily in oncology, and what Horizon are doing in terms of providing, using our gene engineering technology platform to provide the highest quality isogenic disease models where we can engineer models which have knock-ins, knockouts, and point mutations in the endogenous gene. So it closely mimics the way cancer mutations manifest themselves in patients. But also to use that same gene engineering technology to support and accelerate the design of reference standards in specifically diagnostics. But in addition to that, what Horizon are doing in terms of de-orphaning the cancer genome, we have a program in place where we are going after 50 different cancer genotypes over the next 3 years. Some of you may have already heard that Horizon has successfully partnered 2 programs to AstraZeneca at lead optimization stage for first-in-class kinase targets. And in addition, we have several new programs with novel targets coming out in the next year. We live in an era which is post-translational genomics. There's a lot of genetic information out there. What do we do with that information? How can we use that information and make an impact in early stage drug discovery and better outcomes for patients? So this is a challenge. Horizon has been able to access a variety of technologies. The RAV technology is a technology platform which was initially pioneered by the NIH with $50 million worth of funding. Horizon has worldwide IP rights to the use of RAV for gene engineering. We have also in-licensed nuclease technologies such as zinc finger nucleases and CRISPR. We are the only company in the world that has licenses to use CRISPR. And I will be talking about how we use all these technologies for the design of isogenic disease models and their implications in drug discovery. You can see that in the area that once we've generated these isogenic disease models, we look to design and work with companies for example, doing combination studies, where we look at different drug combinations, but also being able to design the world's first genetically defined reference standards. One of the biggest challenges which is faced in the industry in terms of diagnostics, specifically in genetic testing in CLIA labs, is proper controls and standards for IHC, for FISH techniques, for example, and also for pathologists. So Horizon has pioneered this approach and is working with the College of American Pathologists, as well as a number of proficiency schemes around the world. We have over 700 hospitals that participate in this program, and we have a lot of success in providing these diagnostic reference standards, both as part of kits for companies like Roche Diagnostics and Agilent, but also as part of reference standards which can be accessed by hospitals directly. The disease models that we create using RAV is a relatively inert virus. 4 out of 5 people in this audience will carry this virus. We use the virus primarily as a guided missile. We're able to create knock-ins, knockouts, and point mutations, as I said in the introduction, at the endogenous gene, not overexpressing the mutation. So you're looking at physiological levels of expression. And we use the gene engineering to create a whole series of products. These are the various technology platforms for gene engineering that we have access to. Our proprietary technology, RAV, but we've also, as I mentioned in the intro, in-licensed zinc finger nuclease from Sigma, and also we have the strongest position around CRISPR, which is a new technology for gene engineering. And this slide aims to summarize the tremendous work that we're doing at Horizon, and also the various business models that we use. So if you go on the left-hand side, in essence, Horizon is a gene engineering company. We have the capability of working with pharma and biotech partners to create their models of choice for their specific mutation. We have a capacity to generate 200 models each year. We're also involved in a number of consortia, primarily in colon cancer, where we're looking to understand the underlying mechanisms of resistance, for example, as part of Colteras. We're also part of a major FP7 grant in renal cancer, which involves over 200 researchers focusing, again, understanding the biomarkers and underlying resistance mechanisms for renal cancer, and also stem cells. So the next part you can see is how we use these models. Importantly, these models are now being used for patient stratification. So we can actually profile compounds which are in hit-to-lead, lead optimization, or preclinical, or even entering phase 1 to better design those studies, or even what we call target fishing. There's a number of good compounds out there with low tox liabilities, good efficacy, but sometimes the target is unknown. And this is where Horizon can actually help and support. And we have many ongoing collaborations where we do a lot of drug repositioning and drug reprofiling. So this brings me on to the last part, which is primarily in diagnostics. In the diagnostic arena, the biggest issue, as I said, is around proper controls and standards for various techniques and platforms. And this is where Horizon is making great strides in providing genetically defined standards to feed that platform technology, whether it's IHC, or FISH, or PCR. So many of you probably know the ERESA story. ERESA, if we were around when ERESA was developed, we could have helped AstraZeneca in the very difficult path it had through the clinic. This is a retrospective experiment that we did within Horizon. We took all our cell lines and we tested ERESA across our whole panel, and we recapitulated the data that they saw in the clinic. That ERESA was only sensitive to those patients, or in our case, isogenic cell lines, which carried the EGFR mutation. Interestingly, once you added in a secondary mutation, it reverted to resistant. And this is more of our understanding about cancer. Cancer is a disease of multiple mutations, not just a single mutation. This is what we're doing around colon cancer. So this is a part of Colteras and around Erbitux. As many of you know, colon cancer is extremely challenging. Many patients that carry the KRAS mutation will not be prescribed Erbitux. And we found, as part of the Colteras consortium, that if we engineered several isogenic disease models carrying the KRAS mutation, both in the G12 and G13 codon, and we found that G13D isogenic disease models were sensitive to Erbitux. And this data was recapitulated in the clinic. So there is now a design to do a broader study whereby patients that carry the G13D mutation can benefit directly from the use of Erbitux. This is an example of some of the major collaborations that we've had. I'm very pleased to point out that we'll be adding a number of Japanese clients to this list very soon. We also have a number of centers of excellence around the world, one of which is the National Cancer Center in Tokyo, and also the IEG Medical Center, where we go out and teach our gene engineering technology as we work in many different areas, not just in oncology. And in terms of what we're doing in de-orphaning the cancer genome, we use the principles of synthetic lethal screening to do siRNA screening. We use our isogenic disease models and also our understanding of 3D and tumor biology and tumor microenvironment to be able to help identify siRNA hits, which are then validated using our gene engineering technology to create kinase-dead, activity-dead versions, which can thereby better validate those targets. And this is our key program, HD001, which was partnered in March of last year to AstraZeneca. This was primarily in KRAS biology, and we partnered this out in lead optimization. And as I said, we have a second program that was partnered to AstraZeneca at the beginning of January of this year. And so finally, this is as many companies are here in the diagnostic area, and I know we work with several of them. This is an area where I think Horizon is making a real impact in terms of supporting the better design and better patient outcome for kit manufacturers, and also the use of these kits in clear labs as well. So I'd like to finish on this last slide, which really captures what Horizon is doing. First and foremost, we use our gene engineering technology to create models. We license those models to pharma and biotechs and also to academics. We also have generated the world's first endogenous reporter kits. In addition, as I said, we are looking to identify novel targets which are druggable, and as well as being able to collaborate with alliances where we can benefit our customers in terms of understanding patient stratification. So thank you very much for your time.